we're going to go ahead and get started. I'm Steve Roth, Dean of the Graduate School. I am so excited to have 2 hours to just spend time actually outside of the chaos of the moment of graduate education and focus on really why we're here. Graduate students doing awesome and amazing things that will impact society in awesome and amazing ways, and we get to celebrate and value you for the next couple of hours. I'm super excited for that. For all the student presenters, congratulations. You've already actually succeeded in achieving the college campus level finals. You made it through your college competitions. Let's give everybody a round of applause just for that. This is a safe space. Have some fun, right? You've got the story, you've got it down, you've got your 3 minutes. Yeah, the 3-minute thesis competition, we've been doing this on campus for close to 10 years now, something like that. We have had international level winners come out of this competition, but we have always had exceptional presentations. And so really I appreciate all of the organizers of this event, the folks who are staffing and supporting the IT. This is being live streamed out there. I want to thank the faculty judges in the back. I don't envy them having the difficult task of identifying the best of the best in this. And I'm grateful to our MC, Marcia Gensler-Stevens, the Executive Director of the STAMP Student Union, who will actually lead us into the presentations. And you don't want to hear any more from me. Let's get to the presentations. Let's do this.
Well, today I just left my office and I said, I'm going downstairs and I'm going to be brilliant when I come back, because for the next 12 months I will quote from you, because I still remember stuff from last year. So it's a great day to feel brilliant as a result of our students. So let me tell you a few quick things. One is I want to remind you, as Steve did, that we're live streaming this. And it'll be on YouTube later today on the 3-minute thesis site. So you can send that to grandma or grandpa or whoever it is that might want to similarly be educated with me. For people in the room today, you will see that there's some hard copies, not everywhere, but in some locations with the name of the presenters as well, again, suitable for framing. So you may want to take one of those. And then there's a website at the grad school that has, that's listed at the bottom of that print program as well. It'll list the finalists, right? And then it'll list the YouTube. So again, great for transferring to folks in your life who might want to see some of this great knowledge, and or you might want to watch it again. As Steve mentioned, these are not just your average 3-minute thesis people. These are the finalists. We have gotten through all the other portions of the competition. And so this will be just an extraordinary day where it's fast and it's beautiful and it's all over the map in terms of how we make knowledge. I am going to introduce the folks in the back of the room. They have moved up in the world. They have a table, but they're way in the back of the room. So turn around so you can see them, because they're going to make some brilliant decisions. Zina Zakaria is an associate professor and director of International Education Policy Program. Her first love is science. Thank you so much, Zina. I was an undergrad biology major. I appreciate that, before she discovered education as a field of research and practice. Larry Washington is a professor of mathematics and also the undergraduate chair of mathematics. He's been teaching at Maryland for 48 years. Outside of math, he is in an age group track meets and plays bassoon in the community orchestra. So anybody doing something on music or track, you go, speak to him. Isabella Akanyis is a professor of government and politics and associate dean of graduate education for BSOS College. Her research is in the intersection of environmental politics and social inequality. And outside of academia, she's a proud commissioner of the College Park Restorative Justice Commission for the Lakeland community. So both community and the work she's doing in her field of study. And then finally, Chris Bender is an associate dean for communication in the A. James Clark School of Engineering. He's been fortunate to work in communications for more than 20 years. and teach graduate level courses on that subject. He's been a professional speechwriter, we could all use one, on Capitol Hill, maybe not there, and for the former mayor of Washington, D.C., and he helps make really compelling stories from the people he works with and make sure they have the confidence to tell those stories. So we are going to see some incredibly competent, bright people today, and I hope you all enjoy, but we know that our judges are going to be really doing exceptional work. They will select, just so you know where we're headed, they will select 6 winners, right? And that'll be made, that decision, because there's lots of deliberation, I'm sure, will be made on the 3MT website on Friday. And winners will receive $1,000, which is good these days, plus the professional production of their 3MT presentation. So the winners will be known on the 18th. So I'm going to introduce each of the presenters. They will present, and they will do their thing. We'll take a deep breath as they sit down, and I'll introduce the next speaker. And that gives everyone a moment to just sort of think about what they just learned, and for the judges to take a moment to take copious notes about the presentation. I'll also intersperse between those introductions a chance to know a little bit more about what the heck is a 3-minute thesis competition. So we'll add that in when we are giving people a chance to catch their breath. This first segment are all students who are in the master's level in this first, am I right about that?
Actually, it's the first 6 that are masters or pre-candidacy.
Perfect, pre-candidacy, any pre-candidacy. So your pay is slightly less than the people after. The first 6, all right. So this means they're brilliant, but they're not paid as much as the people that are coming later. That candidacy gets you some money. So you're gonna get a chance to meet each one of them, but I start today with Karoline Trovato. Karoline Trovato is in psychology, and the title of her 3-minute thesis is Educate and Empower, Evaluating an Intervention to Empower Women in Future Romantic Relationships. I'm ready for you.
Thank you. First comes love, then comes marriage, then comes women doing twice as much housework and childcare as their male partners. No wonder women consistently report being unhappy and more stressed in their relationships than men. Women tasked with most of the family work lose earning power and struggle to advance in their careers. This is not the future we want for young women, and it doesn't have to be. In the effort to prevent women from shouldering the majority of family work in their future relationships, I developed the innovative Partners Intervention video to educate college women about family work distribution, healthy relationship communication, and a model of effective communication. The intervention also was designed to improve women's confidence communicating in a relationship and empower them to make thoughtful choices in selecting a future partner. But could this intervention really equip women with the knowledge and confidence needed to establish an equitable relationship? To answer this, 303 cishet women completed a pretest questionnaire, were randomly assigned to one of 3 groups, the partner's intervention and 2 controls, and then answered the same questions again. In analyzing the data, I found that the partner's intervention was effective in educating college women about family work distribution, effective communication, and the partner's communication model. It also improved women's confidence communicating with a partner about family work. But could this intervention really empower women in the way that we wanted it to? To find out, I conducted a follow-up study to understand the specific qualities young women seek in a partner and if this changes based on the partner's intervention. Women who did not complete the partner's intervention wanted partners who were funny, attractive, or sociable. One participant even reported nice teeth and lips as a most desirable trait. On the other hand, women who received the partner's intervention prioritized family work and communication-related traits. One participant wanted a partner who was willing to communicate directly and expects an equal relationship. Others emphasized qualities like understanding, empathetic, and a good listener. These findings have powerful implications for young women's future relationships and careers. The partner's intervention could educate women about family work and communication, and empower them to select equitable romantic partners. These findings have powerful implications, and now we aren't done, right? We need to also see if there are follow-up interventions that we can do to improve women's confidence and relationships. We need to find out if, we can understand if men, wanting to empower men to understand the impact of unequal family work distribution and assess women and see if they can understand the importance of their partners in future relationships. It is my hope this research shapes future interventions to increase relationship satisfaction, reduce depression, equalize family work distribution, and advance vocational success for women, paving the way for a more equitable society where women can truly thrive. Thank you.
Thank you so much, Karoline. I have to tell you, I teach a women in leadership class, and I was just thinking, oh my gosh, Karoline should come to class. Our next point person going to share something of his research is in chemical and biomolecular engineering. Rajan Jayasankar is a person who's going to talk to us about something really interesting as well. The residence of vaginal microbiome. This sounds really interesting. Take it away.
Thank you. So, I was watching Netflix the other day when I got an ad, as Netflix does these days, and the ad was for a probiotic supposed to improve your digestion. Now, whether that is true or not, I don't know, but it got me thinking because people understand now the importance of the gut microbiome, but they overlook the fact that there's lots of different microbiomes in the human body. And I think it's useful to define what a microbiome is. It's a group of bacteria that have a mutually beneficial relationship with our bodies. For the gut, they get some food, and we get increased nutrient absorption into our bodies. And at the Jiannan lab, we're interested in the vaginal microbiome. So the vaginal microbiome is really important to protecting the female reproductive tract and making sure that a woman can stay healthy, and it also impacts reproductive and neonatal outcomes. We know that, and of course, everybody's vaginal microbiome is different. We know that depending on the bacteria present, that the activity of the vaginal microbiome is very different. And I like to think of the microbiome as a community. And as any good community planner, I want to know what kind of bacteria are present, how fast are they moving, what are they doing, and really what I'm interested in is what are they making. We know that the bacteria send out different signals to the bacteria around them, as well as to the tissue in the female reproductive tract. Now, these signals can vary based on what's going on in the host body. And at the Jiannan lab, we're interested in what happens when the patient is pregnant. We've shown that the signals sent out can change based on the pregnancy and what the stages are. And by examining these signals, we can predict things like risk for preterm birth and gestational diabetes. Now, and like I said, everybody's vaginal microbiome is different. So I'm interested in examining how different compositions of bacteria affect the signals put out, how that affects mom, and of course how that affects baby. And using this information, we want to leverage a new field of personalized healthcare and improve reproductive and neonatal outcomes for women all over the world. Thank you.
You just gave me something to talk about over dinner tonight, Rajan. For some reason, I feel compelled to talk about stuff like this, and my family's like, ooh, no, okay, great, super. You'll notice that the first two presenters each had one slide. You might wonder, why just one slide? Wouldn't it have been cool to see other slides? Yes, it would have been, but the rules of the game say at the University of Queensland, you can have one static slide, which is so beautiful for those who grew up like I did with overhead projectors, right? This is like old time. So thank you for those fabulous one slides. You'll really appreciate them even more now. The other thing I would say is that you may want to talk to some of these presenters because you have an interest in, the vaginal microbiome, right? Or you have an interest in how to maybe take that test and take it home to talk about shared work. You can do that because we're going to take a halftime break and there's no marching band, but there is ice cream and tea and coffee. So I hope you'll take time to be in conversation with each other as well. So, are you feeling it? You ready? Beautiful. Hanjin Lu is an amazing person in plant sciences. She is pre-candidacy. And the topic today is strawberry savers, ending the fungicide gamble against gray mold. My refrigerator has one of these gray mold strawberries in it. So take it away.
Imagine your favorite potted plant withering overnight, covered in mold. You search online for remedies and spray chemicals. It works. But what if you use the wrong medicine? Unfortunately, you watch it die. That heartbreak? Now, what if I told you that strawberry growers actually face it 1,000 times a day as they battle a major disease, gray mold, caused by botrytis? A fungal pathogen which can turn the vibrant ruby red field into a rotting wasteland in just 1 week. Growers once effectively controlled gray mold using fungicides. However, many of these once reliable treatments have become ineffective over time as the pathogens have evolved to be fungicide resistant. Why does the resistance occur? The primary reason is genetic mutations in the pathogen. Fungicides are like keys designed to feed specific locks on the fungus to disable it. But when the fungus changes its lock through genetic mutations, that key no longer feeds. As a result, growers are left to guess which fungicide might still work and randomly try some fungicides, then hope for the best, just like gambling. And unfortunately, with resistance level rising year after year, growers frequently find themselves on the losing side of this high-stakes game. It's a heartbreaking reality. We don't want disease management to be a losing gamble. What we want is to use the right fungicides to effectively control the disease. That's where our research comes in. In collaboration with extension specialists, we collected strawberry samples from various farms before the season started. By growing these samples on media mixed with different fungicides in the lab, we identified their resistance level. Then we share these results with growers so they know exactly which locks are present in their fields and which keys or fungicides to use. Like receiving a prescription from a doctor. They are thrilled with the results. But we are not stopping there. While helping growers pick the right keys for today's locks, we're also analyzing the pathogen's resistance across different years, regions, and production systems to develop smarter fungicide spray programs. In the future, we plan to apply DNA sequencing and genome editing to deepen our understanding of their resistant mechanisms. Our goal is to make every spray count. So, no more guesswork, no more wasted sprays, just strawberries safely making their way from fields to your plates. Thank you.
Hanjin, that I always think it's really cool when you figure it out and farmers are happy, right? There's not that a lot that we do sometimes that really responds well to farmers and fields. And strawberries are totally critical this time of year. So thank you so much. All right, besides the one slide rule, the other rule for the 3-minute thesis, all of this coming from Queensland, and they have great senses of humor, no poems, no raps, no props, and no costumes. So don't expect any of that in this performance. If we do, disqualified. Not really, maybe, I don't know. But I am thrilled to present our next presenter. Grace Phelps is also pre-candidacy in criminology and criminal justice. And the topic you'll hear Grace talking about today is how close is close to home? An analysis of the Uvalde active shooting and emergency preparedness. A critical topic, and we're glad to have you here, Grace, to talk about it.
Good afternoon, folks. I want to start off by asking a question. When it comes to active and mass shootings, how close is close to home? For many of us, students and professors, active shootings likely fall incredibly close to home for many of us. Despite the fact that active shootings make up less than 1% of total homicides, these attacks are devastating, and researchers consistently show that there are severe negative mental health effects of both direct victimization, but also through media exposure. However, researchers have yet to look at whether this severe negative mental health effects manifest as future emergency preparedness. This becomes increasingly important as we see more and more high-profile attacks in national news media coverage. Take the Uvalde active shooting, for example. Despite the fact that police responded on scene to the school within minutes of the shooting occurring, police remained outside the door of the room that the shooter was known to be in for over 77 minutes. This prompted national outrage and prompted me to consider how might individual level emergency preparedness change after people see that their local government and local police failed to step in on their behalf. So with this in mind, I wanted to look at how emergency preparedness and active shooting risk perception changed after exposure to the Uvalde active shooting. The initial results are in line with what we may think. General preparedness and risk perception both increased after exposure to the Uvalde active shooting. However, like I said earlier, the question that I was really interested in asking is how close is close to home? I wanted to measure closeness in two ways. First, through physical proximity. I wanted to see if individuals who were physically closer to Uvalde, Texas, the site of the shooting, if they were more likely to prepare or perceive active shootings as a risk. And this is what I found. They were more likely to prepare. However, there wasn't a relationship with risk perception. The second type of closeness is what I really wanted to talk about today, social proximity. I wanted to look at Hispanic or Latino respondents, given that the Uvalde, Texas community is predominantly Hispanic. So I wanted to look at how seeing victims of an active shooting who you share an identity with, whether that prompts differential responses. As you'll see on the bottom of my slide, Hispanic or Latino respondents were 7% less likely to perceive active shootings as a risk prior to the Uvalde shooting. However, after seeing individuals who they share an identity with attacked, their risk perception was 6% higher after the Uvalde active shooting. So ultimately, how close is close to home? It looks like distance does play somewhat of a role. However, my results indicate that we should be looking more towards social proximity to see how certain individuals who share an identity with national news media coverage victims may be more likely to prepare for future active shootings. Thank you.
Thank you so much, Grace. I appreciate that, and I think a lot about your results, because I think about how much we changed as college communities after the Virginia Tech shootings, right? And we started to think anew. All right, so origin story. The 3-minute thesis. It starts as a competition in Queensland. Now, they were suffering a drought. Kind of, Maryland right now is in a drought, not as bad as Queensland was. And to conserve water, you were required to not take showers longer than 3 minutes, right? So you stood in the shower and you recited what you had learned from your thesis or dissertation. It got you ready for the day, maybe. But that's where it all started. And so they decided, why not have a 3-minute thesis or dissertation presentation, because we could rehearse while we're in our showers. So we're glad to be a part of it. And yes, we have had winners, as Steve said to you earlier. Hannah Burchard is going to present, and she represents Animal and Avian Sciences as a degree place. She is also pre-candidacy. And what I love about this is she's going to talk about a shorter route to quality dairy production. And I have a brother-in-law who grew up on a dairy farm, so I'm listening.
saying goes, good things come in small packages. But how accurate is this? Let's put it to the test. My name is Burchard, and I study short stature corn. Short stature corn is a new variety of corn recently introduced to the dairy industry and is suggested to be efficient in growth size, harvesting, and ensiling compared to conventional corn, or what is traditionally used by dairy farmers. Corn silage, which is the fermentation of the whole chopped corn plant, is a key component in dairy cattle diets. It comprises of about 40 to 60% of forage on a dry matter basis, which means quality is essential when determining which forage source to use. Now, that's the source. How do we ensure that the silage is stable as it's fermenting? The answer being organic acids. Organic acids have been studied and found to reduce yeast and mold growth, preserving the stability of silage as it ferments once it's inevitably exposed to oxygen. Now, how do we put all these different components together? Let me walk you through our study. With the help of our partners at the Central Maryland Education and Research Center, we were able to analyze both short stature and conventional corn at time of harvest, as well as create our own experimental units for our own analysis back at the lab in what we termed experimental mini silos. At time of harvest, we were able to look at nutritional factors to compare both varieties and found that they did compare in nutritional value, which is promising because they are a key energy source to dairy cow diets. Now, back to those experimental mini silos. In our lab, we were able to analyze the fermentation profile over 6 different time points, that being day 0, 7, 15, 30, 60, and 120. Based off of our results, we were able to find that the fermentation profile, as well as the silage stability, was improved with the application of organic acids by reducing those yeast and molds due to the production of total acids, and then preserving the stability of silage. We continue to investigate this new variety because of its potential to be a high-quality forage source. Our goal of our studies always come back to the farmer. If we're able to find a quality forage source that does not require new equipment or drastic changes to management practices, this lends to the sustainability of our forage source and the effectiveness of the product. This is great news for farmers and great news for quality production. Thank you.
Thank you so much. All right, we're going to do a quick quiz here. Did anyone ever grow up near or in a farming community? Anyone? Oh, look at this. Look at this. You have fellow farmer / people who might have done research or just lived close by. Here's the interesting thing. If you were a student at the University of Maryland at its founding, well, you'd be male, but everybody had to work on the farm. It was required. Right? So we would have been looking for the best corn crop to grow on McKeldin Mall, or what was now McKeldin Mall. So how fabulous is that we all would have claimed to be farmers at a much earlier juncture. I'm so grateful to you, Hannah, for what you shared with us about your research, and keep going. We need you. We are going to now shift gears again back to our friends in chemical and biomolecular engineering, our last of our pre-candidacy candidates, Alyssa Petersen. You're going to see sort of an interesting kind of juxtaposition of a fellow chemical and biomolecular engineering student, developing an in vitro platform to model cross-kingdom communication in the female reproductive tract.
We've all been stressed about work, about exams, about papers. And whenever you're stressed, you have different ways of showing it. Some of us might get angry and loud, or quiet and seclude ourselves. Maybe you eat too much, or maybe you don't eat enough. Any way that you do it, you're telling everyone around you, hey, I'm stressed right now. And each of those different ways of saying I'm stressed has a different impact on the people around you. Maybe you snap at a friend and you push them away. Or maybe your behavior becomes so concerning that your family starts to pack your lunch and check that you've eaten it. Any way it happens, your signals are changing how the people around you are acting. If we scale that down to the cellular level, it's no different. When you're stressed, your cells are stressed. And they're sending out signals to everybody around them to let them know. Most of those cells getting those signals are going to be bacteria, because we have more bacteria in our body than we do mammalian cells. These form microbiomes, and they have a direct impact on your health. we're particularly interested in the vaginal microbiome and its impact on reproductive and pregnancy outcomes. It can prevent or reduce sexually transmitted infections and diseases, and it has a great impact on pregnancy outcomes. So when you're stressed, right, your cells are sending out signals. One of those signals is cortisol. That sends a fight or flight response to all of your cells. So they stop focusing on what they're typically making, which is going to be your vaginal mucus, which is where these guys reside. That's what these bugs live on. And so their community, their environment is changing, and now they're going to start sending out different signals. But why do we care how their signals are changing or our signals are changing? Think about why a therapist cares how you act when you're stressed. They need to understand what you're saying, how you're saying it, and why, in order to help you develop more productive and protective ways of dealing with your stress. We want to do the same thing. If we can hijack these natural communications that the bacteria have to make these signals more protective and productive, we could possibly prevent sexually transmitted infections, preterm birth, preeclampsia, all of those different negative outcomes, and create a healthier environment. And once we figure out how to do this in the vaginal microbiome, we can extend it to other microbiomes, like the gut for colorectal cancer, the eye for glaucoma, the lungs for pneumonia, and so much more. Thank you.
Wow, right? So far, you've learned about so much. In this case, I'm just thinking about my stressed out cells and where the heck they're going now. But how darn interesting. And I think about vaginal mucus. We don't talk about that all too often, but we probably need to talk about it more. And definitely something that might alleviate sexually transmitted diseases. Thank you so much, Alyssa. Our next presenter begins the long line of post-candidacy presenters. And we are so lucky to have those brilliant souls with us. Tifanee McCaskill is a student in TLPL, Urban Education in the College of Education. And she is going to make her dean, who I wish was here, really happy because she's talking about teacher recruitment. But this case, she's talking about Black teacher recruitment begins in kindergarten. Tifanee, welcome to our world. You're going to be great.
All right, here we go. We need Black teachers. Black teachers bring unique experiences, perspectives, strategies that would otherwise be lacking in schools. So we all need Black teachers. But currently in the US, only 6% of teachers are Black. That means that most people will go through their entire K-12 education without ever having the opportunity to learn from someone who is Black. Now, it's clear that we need Black teachers, but what's unclear is the best way to recruit Black teachers. And I believe that Black teacher recruitment begins in kindergarten. See, there was a time in the US where teaching was a popular profession among Black folks. But currently, countless school districts are struggling to racially diversify their teacher workforces. And I think it's because they're spending all their energy trying to convince Black STEM majors or engineers to just abandon their six-figure paycheck and become a teacher. They're trying the same thing over and over again, and it's just not working. We've had a teacher shortage since I was in elementary school, and I'm years old. Like, I think we're missing a very large piece of the puzzle, and that is Black teacher recruitment begins in kindergarten. I often ask adults, why do you think Black kids don't want to be a teacher when they grow up? And usually their responses have something to do with low teacher pay, which you would expect from an adult. But do we really think that little Sally in her ELA class is worried about her 401k? Like, do we really think that 12-year-old little Billy in pre-algebra doesn't want to become a teacher because he's worried about how he'll feed his wife and kids on a teacher's salary? Or do we think that maybe their interest and disinterest in teaching has more to do with their experiences in schools and with teachers? See, the unique thing about teaching, it's the only profession that students experience up close and personal every single day. When you think about it, children are learning what it means to be a teacher through direct observation every single day of their life before they make a decision. right? And so the reality is that a lot of black students are going through 13 years of school and at the end of that deciding, nah, that's not for me. So my research seeks to understand the reasons black students don't want to become a teacher when they grow up and what the public school system can do to change that, right? Like, what are these kids observing that's making them decide, no, teaching is not for me? Because how can we create effective solutions if we don't truly understand the root of the problem? See, unfortunately, most research around the black teacher pipeline does not center the voices of black students, right? Like, most research, since it doesn't center the voices of black students, we never know why black students don't want to become a teacher. So, hear me clearly. If we want to just throw scholarship money and higher wages at students who don't want to become a teacher, it's not going to work. The best way to recruit black students is beginning in kindergarten through their relationships with teachers and in schools. If we want to create relevant and equitable pathways to teaching for black students, we have to begin by elevating the voices of black students so that we can truly understand how their experiences in schools impact their decisions on whether or not they're going to pursue teaching as a career. That's it.
As you were talking, I was sitting there thinking, I can still tell you about my kindergarten teacher, right? And for some of us, that was a pivotal moment in life. I was a little crazy in kindergarten, but it was good, and she was understanding. Think about how our work as researchers influences the future too, and you saw that just now in the work of Tifanee. Our next researcher is going to talk to us about something. If you saw Maryland today, you saw that Gail King went up in space, scared as she was of flying, and there were pictures that were taken from there. I think of that when I look at Yiming Zhang and the work that they are doing in geographical sciences. The topic is Seeing Blue from Space, the long road to recovery in Puerto Rico after Hurricane Maria. And we're so glad to have you here.
Oh, it works. Good afternoon, everyone. Now imagine your roof got ripped off in a hurricane, and the government hands you a blue plastic tarp and says, here, this should last 30 days. And then imagine that same tarp is still your roof. 6 months later, a year later, or even several years later. That's what happened in Puerto Rico after Hurricane Maria hit in September 2017, damaging over 90% of homes. The government, including FEMA, handed out thousands of blue tarps as temporary roofs. But there was no system to track it. That's where I came in. I want to figure out where were those blue tarps and how long they last. And I thought, wait, we have satellites flying over us all the time, right? What if I used the satellite images, like eyes from the space, to track those blue tarps? I analyzed 5 cities across Puerto Rico by using these satellite images. And what I was found was pretty surprising. In most places, those blue tarps stayed up at least 9 months, sometimes even more than a year. And they were not randomly placed, mostly in low-income and vulnerable communities. That was a big question. Why? Why is this recovery taking so long? So I went to Puerto Rico for fieldwork last summer and talked to people face-to-face. And honestly, it was eye-opening. There were still homes with blue tarps even after 7 years. And many of them are in low-income and vulnerable communities. One of the biggest reasons is that, owing to a historical problem, the buildings were built informally, so no paperwork, no legal ownership document. So when people apply for help from FEMA, they were told, sorry, we cannot help without proof you own the place. And many people I surveyed with were elderly, without cell phones, without internet, totally disconnected from the system, needing to support them. So my research mainly fills in these gaps in two ways. First, by using satellite images and fieldwork to see what's happening really on the ground after disasters. Second, by pushing for recovery system and to make it more inclusive and just. No one should have to live under a tarp for years. My research makes these invisible struggles visible, not only in Puerto Rico, but anywhere where disaster strikes, but recovery falls short. With the right tools, with the right priorities, we believe we can see better, do better, and recover better for everyone. Thank you.
Thank you so much. You can see a lot from satellites. They're probably watching us right now, which is so totally true. I was with my 5-year-old great niece in Chicago last weekend, and she is a preschooler. She'll start this fall, she told me, in school. So someday she'll be a teacher because of what happens in kindergarten. And she told me that she had to do a paper on porpoises. I don't even think I knew how to read at that age. But she drew a very nice picture of a porpoise and wrote some things. And one of the things she taught me is that porpoises, when they are hungry, they have to sort of jump up and hit a fish before they eat it. She said, it's called fish whacking. And I said, really? It's called fish whacking? And she said, look it up. And so fish whacking is our new favorite phrase. And I say that because our next presenter won't be talking about fish whacking at all. But you could imagine fish whacking in the world in which we're about to learn. Now, I have a bet right now that I will pronounce this absolutely correctly. Smrithan Ravichandran, did I do it? Not so fast, but close, is in chemical physics, which I like that combination a lot. And he is going to talk to you today about harnessing powerful lasers to help build a fish tank of the universe. And there'll be some fish whacking in that fish tank. So, Smrithan, come on down.
What do you do if your kid was very curious about little fish? To avoid all their questions, you might buy them a fish tank. Bring home a small slice of the vast ocean that they can learn from. But what if they're curious about the vast empty space in our universe? It turns out that there are some secrets hidden within the vacuum that makes up most of our universe. So wouldn't it be nice if you could bring home a slice of intergalactic space? Well, a bunch of these curious kids grew up and became scientists that built magnificent tools to learn about our universe from within a laboratory, like lasers that can make a short flash of light stronger than the average power of sunlight across 200 billion football fields, focused to a spot as wide as your head. And these lasers can help safely replicate the extreme conditions of outer space inside a lab. But using these lasers in a precise manner to study fundamental properties of the vacuum has a couple of problems. Number one, we need to know exactly how strong the laser is when it is focused to a spot so intense that no material can survive. Number two, we need a near perfect vacuum when there is no way to remove every single particle from within a box. To tackle these problems, I study how tiny particles called electrons move under the influence of the world's most powerful lasers. I evacuate as many atoms and molecules as possible from a chamber and focus a laser to a spot that rips out electrons from these remaining atoms. I showed that for increasing laser intensity, these electrons are ejected from the laser focus closer and closer to the direction in which light travels. So we can find out how strong the laser is by measuring the ejection angle of these electrons. We also found that imaging these electrons shows us how well the laser is focused when we can't directly stick a detector in that spot. Now, getting rid of the electrons still gives us a cleaner environment to study the vacuum, even if we can't get rid of all the particles. So I used a second laser pulse to see how well these electrons are cleared by the leading laser pulse. Sort of like shooting two cue balls on a pool table. One to clear and another to see what was left behind. I found out how long it takes to create a less cluttered environment for vacuum-based experiments. And these findings play a crucial role in allowing us to harness the world's most powerful lasers for a variety of phenomena, from revealing the secrets hidden in empty space to enabling fusion energy and radiation therapy. But it is my hope that someday I can help you answer all the curious questions that your kids may have, thanks to the fish tank of the universe that we can make in our laboratories.
Thank you so much, Smrithan. I really appreciated it. I'm going to go home with some really good stuff, and I'm calling a 5-year-old. You think you know everything. Yan Lee is our next presenter, a student in physics. She is going to talk to us about cats, dogs, and electrons, and how ferromagnetism emerges. This is going to be good. More to talk to the 5-year-old about.
All right. Guys, we're so close to ice cream. I'm the last one speaking before ice cream, so just bear with me. Imagine there's a town filled with cats and dogs, and every house can hold up to 2 pets. But there are some rules to it. Cats refuse to share a house with another cat. They just won't. A cat and dog can share a house, but it's kind of a hit or miss. Sometimes they get along, but other pairs would fight all the time, making life super stressful. So what happens when they don't get along? Well, these cats aren't stuck in one place. They're constantly moving from houses to houses. But if they kept going to houses where they're forced to share with the opposite type, there's chaos and stress. So they figured a better way. If they stayed close with their own kind, they can move freely and happily, and hence creating a cat neighborhood and dog neighborhood in town. This is exactly what happens with electrons, tiny particles inside materials. Electrons also follow rules. 2 of the same type, which we call spin, can't share the same spot, much like how cats refuse to share a space. Opposite spins, if you're forced them together, costs a lot of energy. Similarly, electrons aren't stuck in one place, they're constantly moving. But if different spins of electrons are just randomly mixed, their movement becomes difficult, requiring extra energy, just like the extra energy you'll need to clean up the mess of forcing cats and dogs together. On the other hand, if electrons of the same spin start to group together, they can move within themselves, and hence lowers the overall energy. So even though electrons are free to move, they naturally start to group in the same spin, just like how cats and dogs forming their own neighborhood. And this surprising self-organization is one way ferromagnetism can happen. That is the force behind magnets, computers, and many future technologies. Tiny little particles, just like playful pets, ends up creating something much bigger. Now, in my research, I want to understand the rules of this town better. What factors will decide whether the cats and dog will separate? For example, how strong is their dislike to each other really? What if the town has a different layout, like narrow streets or open park? These details, like the strength of repulsion interaction and the geometry of the system, will determine when ferromagnetism happens and when it doesn't. To answer these questions, I've created phase diagrams, which acts like a rule book for the town. By studying this rule, we can know exactly when and how the cats and dog will separate, based on the different condition given. By studying these rules, we can now discover new quantum materials and gain a deeper understanding of one of nature's most fascinating phenomena, ferromagnetism. Thank you.
I think in each instance, while it's only one slide, they're so darn beautiful and creative that they have you thinking about it long after the words have stopped. So thank you so very much, Jan. I want to tell you one quick thing about ice cream before you go eat it so that it's a research experiment. The University of Maryland dairy is more than 100 years old. And if some of us have been here for 40 plus years, they used to make some of the ice cream down in Turner Lab, but now it's made elsewhere, but it's still made by University of Maryland staff. And the flavors downstairs are great. It used to be in Turner, and then that was going nowhere. So they moved it here, which is great. But The other thing you want to know is that vanilla ice cream at the Maryland Dairy got voted the best vanilla ice cream in all of Washington, D.C. But you won't find it for sale on the shelf because it has a very high butterfat content, which is why it's so delicious. So if you're in a need to sort of fatten up a little bit, you eat the vanilla, okay? So we're going to take exactly an 11-minute break. All right, you can have 14 minutes. You can have 14 minutes to get something to eat, including that vanilla with a high butterfat. Oh, you guys are good. You're probably in an ice cream induced coma now, aren't you? But these folks are going to wake you up in the second half. Thank you all for coming back and for listening, and for those of you who just joined us for the second half because you've got someone really special in the second half, welcome. Can we give a big shout out to Robin? And Robin, I know there are other people in the grad school who also make this happen, but I know you really do make this happen. And I am so grateful. And to our judges who are working really fast, because I'm trying to vamp a little bit between, but woo, we're talking fast, and which is great. And Chris, thank you for managing the sound, which is going beautifully and will be recorded beautifully so that you can rewatch any of these that you might be interested in sharing or knowing more about, which is great. We enter the second part. In this whole second part, everybody is post-candidacy, so they could buy you ice cream tomorrow with their new raise. It isn't much, but one scoop. I have to tell you that we are just already on like a head-spinning kind of thing, and I don't know which or how many of these have stuck in your head, but I am already thinking about any number of conversations that I'm going to start later this afternoon. Our first one up in the second half, Sagnik Bhattacharya, is a student in electrical and computer engineering, and you're going to hear him talk about modern data problems that require modern solutions. Come join us.
Hi. Think of a smartwatch for the brain, a small wearable device that can track neuronal activity. So if it has to be small and wearable, it has to track the minimal number of neurons possible and efficiently store and process the collected data. So as developers of such a device, we have to be really, really clever about what data we collect and when we do so. Which raises an immediate question. How do we make sense of a system as complex as the brain from this partial data that we collect? And the answer to that comes from exploiting the structure of neuronal data. So it turns out that neurons in the brain only talk to other neurons that they are connected to. And what this means is that neurons that are closer together fire together more often than more distant neurons. In my research, I use mathematical models called Markov random fields that capture these local dependencies in the form of a network of nodes and connections. The goals of my research are threefold. First, to identify a representative subset of neurons. What that means is, let's say you walk into a room, you listen to just a few of the people so that you get a gist of the conversation. The second goal is to take the data that we collect and learn the relationships among the neurons. Third, I use these relationships to more efficiently compress and store the collected data. Now the cool thing is that even though in this talk so far, I've only talked about neuronal data, such patterns show up everywhere. From robot swarms, to self-driving cars, to even social networks. So in social networks, our activity is more correlated to that of our friends and communities than people that we just don't know. And so you can use insights from my research, if you are a manager of a social media company, to identify sets of influential users or influencers to, let's say, track the spread of misinformation on the network. And something that surprised even me was that I was able to use insights from my research to better understand a problem like secure communications over the quantum internet. Cool, right? So just like smartwatches give you health insights from limited sensor information, my research allows the development of tools that allow you to decode complex systems from the brain to social networks based on limited information. It's a small window into big opportunities. Thank you.
What I love is this snippet that you just listened to. Like 20 years from now, you'll be like, oh, I was there when he started. Like, you know, you're gonna really think about that, right? And how grand it is to think about all of the knowledge you'll walk away with at the end of this. Steve the grad dean alluded to the fact that we've had winners. In 2014 and 2017, our nominees from the University of Maryland in the 3-minute thesis competition won the judges' first place prize and the People's Choice Award. So everybody's a winner, but we've had some big winners. So bravo to Maryland and bravo to all of you who even came to participate. We are so glad to have Benjamin Lanham with us, and he is a student in biochemistry. The title of his presentation, I can't wait to see this because just of how, exclamation mark, stop COVID, no running with scissors. So bring it on.
I want you to think about the last time you were in a store and you saw a security tag on a short. Did you get the intrusive thought that if you had a pair of scissors, you could cut that tag, you could steal that short? Well, a similar conversation is happening in our cells during a viral infection. During a virus infection, all cells produce ISG15 and ubiquitin chains, which act as a security tag that attaches themselves to viral proteins, marking them for destruction. However, unlike our cells in the store, SARS-CoV has a pair of scissors, a protein called PL-Pro, which recognizes these security tags, cuts and removes them, thus enabling the virus to steal our cells. Therefore, understanding how PL-Pro recognizes these security tags is vital if we're going to develop drugs that interferes with this recognition so we can stop the virus from stealing our cells. That's what my research sought to answer. And what I discovered was that ISG15 and ubiquitin chains, despite having slightly different chemistry, are recognized at similar affinity by PL-Pro. Most notably, the main components of the tag bind to the S1, S2 site. In our scissors analogy, we can think of this as the handles of our scissors. Additionally, the tail of the tag makes its way into the active site. We can think of this as the blades of the scissors. Now, this is a great model, except we ran into a problem whenever we extended the tail of the tag, suddenly the handles would not bind to all tags. So this got us thinking, okay, maybe the tail is important for recognition by the scissors. So what we did is we cut back, shortened the tail, and asked what happened. Well, surprise, surprise, suddenly the tag no longer binds to the S1, S2 site. Okay, well, now what we did is we extended the tag, made the tail much longer. Again, we saw a disruption to the binding to the handles of the scissors, but annoyingly, the scissors still cut the tag. Okay, so now maybe we don't even need the S1 site binding for the scissors to cut. So we modified the tag to disrupt binding to the S1, S2 and ask what happens. Well, even though the rate at which the tag was cut was reduced, PL-Pro was still able to cut the tag. What does this mean for COVID? Well, it means that when we are designing drugs to target PL-Pro, we can't just look at targeting the S1, S2 site. We need to go straight for the source, the blades of the scissors, and block that. Only then can we say, stop COVID, no running with scissors. Thank you.
I will never think of COVID the same way, right? Running with scissors, what a great analogy. And the fact that you could cut off the security tag. It's a good way of thinking about science and medicine. Thank you so much, Benjamin. Our next presenter is Jenan Shu. And what I love about the field of study that they're in is decisions, operations, and information technologies, the fastest growing. Oh, and I lied. You're not next. I'm so sorry. We're gonna, you're next. Who's next? Oh, it's your slide. Celia Chen. Chen. Yes, sorry. Celia Chen is next, and then we'll go back and pick up something. And I love that because it's your fabulous slide. The title of this topic is Stethoscopes to Self-Care, How We Decide Who to Believe Online. This person, Celia, is from Information Studies, the fastest growing college as well. I know.
Okay. So, when was the last time you paused mid-scroll to question why you trusted a social media post? Was it the verification badge that convinced you? A professional title? Maybe it was a lab coat in their profile picture. Every day, we make split-second decisions about who to trust online, often without realizing it. And these decisions matter because in the United States alone, 73% of adults, that's 253 million people, are active on social media, with over half relying on these platforms for their news and information. And now, with AI tools like ChatGPT that can generate persuasive content online, these trust decisions have become even more difficult to navigate. Who, or what, should we believe when we're scrolling through our feeds? This is the question at the heart of my research. I study how experts craft their digital personas and build credibility online. My work looks at patterns in expert self-presentation and, in one unique case, how audiences navigate trust with AI. In my first study, I examined professional doctors and how they craft their professional identities on Twitter. While most of the doctors in my study use lab coats to signal their authority, their most effective tweets weren't actually just about medicine. Instead, they combined professional experience with more personal or cultural elements, like one doctor who connected Taylor Swift's Grammy wins to their struggles with electronic medical records. And this humanizing approach let them build trust with both fellow professionals and the public. By contrast, on TikTok, I found that mental health professionals with credentials in their bios actually received fewer likes and comments than those without. And these mental health experts who succeed on the platform rarely talk about their degrees or their research. Instead, they build trust through storytelling, through sharing vulnerable experiences, and by filming in casual settings like their bedrooms, rather than in clinical offices. In my final study, I investigated and interviewed how graduate students use AI for educational support. And I found that students tend to trust the machine with more objective tasks, like code, where a piece of code either works or it doesn't. But they were less inclined to trust the machine when it came to more creative or analytical tasks, tasks that the students considered more core to both their professional and personal identity. And this relationship shows that students are actively negotiating how they feel about AI, rather than just accepting or rejecting it outright. Online credibility isn't just about credentials, it's about connection. And whether you're a doctor who's humanizing your tweets, or you're a therapist who's sharing personal stories on TikTok, or you're a graduate student who's protecting the uniquely human aspects of your work, we're not looking for perfection, we're looking for authenticity. And so next time you find yourself scrolling, pause and ask yourself, are you trusting the expertise or the charisma of the human you think is behind it? Thank you.
Wow. Who knew Taylor Swift was going to be in medicine? Like that somehow she's on, like, my records are so well organized, it's Taylor Swift. Who knew? Now, if did it have music playing in the background of that? You want that. You kind of want Taylor Swift tunes. Thank you so much, Celia. Our next presenter, well, I might have hinted at it earlier. Jiannan Shu is with Decisions, Operation, and Information Technology. And what is the topic of the presentation is, and this is interesting because these all go together, signals in the chat. What drives freelance wins?
Oh, hi everyone. Freelancing is booming, and platforms like Upwork, they make it quite easier to recruit the talent all over the world. Clients make job postings, and freelancers apply. So this word sounds quite straightforward, but here's the thing. Hiring the freelancer with the suitable skills does not necessarily guarantee a job success. Why? Because for a job success, it's really not about what a freelancer can do. It's also about how they communicate along the way. When communication breaks down, then everyone suffers. For clients, it leads to unmet demands, misdeadlines, and misunderstanding, and ultimately wasted time and money. For freelancers, the unfulfilled expectation leads to the the elective experience of working for a client on the platforms, and in the end, it ended into low quality match. So right now, the hiring decisions on these platforms, they rely on freelancers' demographics and their profiles and their rating scores and past rating scores and the previous working demos, but this is just a surface. What really matters is how they communicate along the way. And to study these questions, I analyzed millions of messages exchanged by the clients and the freelancers on the marketplace using natural language processing and machine learning with the objective to understand what are the factors drive freelance success on this marketplace. And here are the findings that I have found so far. So first of all, the consistent engagement really matters. For successful freelancers, they tend to send regular check-ins and share their progress with clients, and they also ask very thoughtful questions. Secondly, for the freelancers' perspective, there was the confidence and reliability in their language also matters, such as they tend to use language to describe their clear commitments and calm tone. They are highly related to the high quality the high quality ratings they received in the end. And lastly, what's surprising is that being too eager, like over-promising, were instantly following up actually result in a bad quality match, which may backfire. So after discovering this pattern in the communications, we can now predict what are the projects that are on the right track and what are the ones that might go off the rails. And this will help freelancers work better and help clients to figure out who are the candidates who don't to be good on the paper, but also communicate very well in the practice. So next time, if you work as a freelancer, you will work with a client. Remember, the job success is not just about the skills you have, but also about the communication throughout. Thank you.
Thank you so much. I am really taking to heart what you were just talking about, because right before I came down here, I was trying to make airline reservations, and I finally gave up on the web and went to a person, and definitely a freelancer. But they overpromised in a very nice way, which was lovely, We are really grateful to have our next presenter, Celia Chen? No? We're going to go with a different one. Emmy Werner is here. Emmy Werner is from Global Environmental and Occupational Health, right? And studying something really interesting. What if we could harvest rainwater, tapping into an alternative irrigation water source, which is fabulous.
Just one urban farm in Baltimore, Maryland produced over 14,000 pounds of produce last year. That is food that is feeding a community. And it's just one of the 33 urban farms and over 200 community gardens that's working in Baltimore. And each one of these farms and gardens is reliant on a water source. But finding a water source that is safe, reliable, and affordable isn't always easy. Just last fall, Baltimore had 38 consecutive days without rain. But during that time, the need for water didn't go away. So this is where we think rainwater harvesting could come in. Because while there was a drought in the fall, in the summer, there was over 9 inches of rain. So if we could capture that water, it could be a really tremendous source of irrigation water on urban farms and gardens, especially as our climate continues to change. However, there are issues with this water. This water is typically collected off of a roof, and anything on that roof, like bird poop, could get into our water. And we don't really know what bacteria could be in that water, especially if it's collected on an urban farm. That's where my research comes in. I'm interested in the microbial quality of harvested rainwater and possible treatment options for reducing contamination if we do find it. So to do this, I have 3 goals. The first, I want to know if there's bacteria in the rainwater that would indicate if there have been feces that have entered it. To do this, I'll be looking for E. coli. Second, I want to know if there are human pathogens in the rainwater. specifically Listeria monocytogenes and Salmonella. And third, I want to know if either a filter or a chemical sanitizer could help reduce the contamination in the rainwater. So I work with a tremendous team to collect rainwater on an urban and a peri-urban farm. We take this rainwater that we've harvested, we set some of it aside and leave it untreated. The rest of it, we either run it through a filter that has sand and recycled iron in it, or we treat it with a chemical sanitizer called peracetic acid. Then we take each one of those water types and we apply it to plots of brassica salad mix. And then we test that water and we test that produce. So what did we find? Well, let's start with E. coli. We found that in the untreated harvested rainwater, there were levels of E. coli that were higher than some irrigation water standards. But both of our treatment options were able to reduce those levels. So what about the pathogens? Well, we found Listeria monocytogenes in about 15% of our untreated harvested rainwater samples, but in less than 10% of our filtered samples, and we never found it in our sanitized water samples. And then when we looked at the produce, no matter what water we used to irrigate, we did not find any of our bacteria of interest. So we think that harvested rainwater could be a really great supplemental source of irrigation, but we really recommend treating it first. Thank you.
Emmy, thank you so much. Did you think about the lettuce you just ate? Or like you were like, oh God, is there listeria on this? You know, like, no. That was really amazing. And I think about the rainwater we had in the last couple of days. Katie Evans is in entomology, which I love that because the first woman who went to the University of Maryland in 1917 studied in entomology. Mm-hmm.
Cool fact.
That's a little factoid there. And Katie is studying, you're going to hear her talk about flower power, how increasing floral resources impacts arthropod communities in cropland. Arthropods. Anthropods sounded like so crazy-ass, but arthropod sounds a lot better. Communities in the world of crop.
Flowers have power, but how do we measure that power? Flowers provide food and refuge to a diverse array of pollinating and plant-eating insects, as well as to many insect predators and parasites. These interactions between plant and insects help facilitate productive and healthy ecosystems. In my thesis, I investigated these plant-insect interactions in an edamame agroecosystem. Natural habitats are ultimately the source of many beneficial insects that farmers rely upon for pollination services and to control pest outbreaks. As farms across the country become less and less sustainable from pesticide use and reductions in natural habitats, eco-friendly solutions are needed. One such solution is to add flowering plants to ecologically impoverished landscapes, i.e. croplands. The goal of my research was to assess the effects of adding flowering plants to croplands on insect spillover between natural and managed habitats, and on the crop and the wild plants in those natural habitats. I carried out a 2-year controlled experiment, and with a team of 13 undergraduate students, we identified about 36,000 insects, counted 5,000 wild plant seeds, and weighed 30,000 edamame pods. So what did we find? Our results are depicted on the slide in the colored boxes and arrows. Adding floral resources increased the abundance and diversity of predators, herbivores, and pollinators. We even found some specialist pollinators visiting our edamame flowers. You would think that increasing herbivores in our florally enriched plots would ultimately impact the damage on our crop. However, yield was unaffected, and in some instances, increased in the presence of these added floral resources. Wild plants that were in closer proximity to these added floral resources experienced greater pollinator visitation and higher seed counts, indicating that these floral supplements will increase the reproduction of nearby wild plants. However, this was not the case for every plant in our study. So why is my research important? Well, recent studies have documented striking declines in global insect populations, and this has immediate repercussions on our food security and food production. My research quantified the power of flowers and showed that adding floral resources to our croplands increased yield and, in some instances, the reproduction and survival of wild plants in nearby habitats. So next time you come across a flower, stop and take a look and try to observe that power for yourself.
It does make you think of doing your part this time of year, like planting gardens and the like, and how critically important it is. I also thought the insects here reminded me of middle school insect collections. Did anyone else have to do that? I think they probably don't do it anymore. It's probably not good, but woo, back in the day. We are so grateful to have Arson Lee with us, and Arson Lee is talking about economics, but in this case, the topic is transit funding fun? A 4-in-1 deal on federal money, which we talk a lot about federal money these days. So bring it on and bring us hope, Arson Lee.
Imagine giving your child money for books, only to see it spent on junk food. That's the essence of the federal government's worry when it gives out grants to cities for education, public safety, or transportation. Even when federal funding is restricted to specific uses, it can displace money local officials would have spent from their own coffers. In other words, more federal money does not guarantee more spending on the right things. I studied this issue in the context of public transit in the US, which is managed locally but funded by federal, state, and local governments. Ideally, we'd run an experiment. Give each city a random amount of money and see how their spending changes. While this isn't feasible, the US government did something very similar in response to the 2008 financial crisis. In an effort to rebuild the economy, they distributed $6 billion in transit funding to large cities with amounts determined by a strict formula. With a little statistical magic, this windfall becomes a natural experiment to study the effect of additional federal funds. I discovered that federal funding was very effective in increasing total transit expenditures. First, state and local governments did not cut their investment when the federal money arrived. Second, and more surprisingly, once those extra federal funds ran out, state and local governments stepped in by increasing their own funding. Altogether, every federal dollar generated almost $4 of transit spending over the next decade. That's nearly $24 billion in new buses, transit facilities, and rail infrastructure. The potential benefits of such investment in terms of lower traffic congestion and better air quality can be huge. In my future research, I want to understand whether this strong response might appear in other contexts, including grants for education, public safety, and health. One reason local governments did not cut their planned spending was the unexpected nature of the extra federal funds. But what's more intriguing is the attraction of new investment. Transit projects launched with federal grants likely helped create lasting commitments to keep the money flowing. The potential for such commitments varies across sectors, and so does the resulting spending response. The main takeaway is this. Federal grants for public transit attract funding from other sources. Even though state and local governments are relatively independent, they respond to the government's carrot. This power is something federal policymakers must consider when deciding where to plant the seeds of new grants or whether to cut down a garden that's already growing. Thank you.
2008 reminds me of a time we might be in right now in the economy world. Thank you so much, Arson Lee. We are so grateful to have Chen with us, a student in electrical and computer engineering. The topic of this presentation is a diamond in the rough, polishing quantum sensing without radio waves. Bring it on.
I've been hooked on the New York Times connections game recently, where seemingly unrelated words come together in surprising ways. Like the words quantum and diamond, there's a fascinating connection we can resolve here together. Invisible waves surround us, like the magnetic fields from the phone you play connections on, or from your heartbeat. Picture a quantum sensor as a ship navigating these waves with exceptional precision, mapping out the data to help us better understand the world. Now, imagine the ship being made of diamond, smaller than my fingernail. No, not a shiny diamond prized in jewelry, but these are synthetic diamonds made with special treatments to create atomic impurities known as nitrogen vacancy centers, or NV centers. So these quantum diamond ships are remarkable because they're robust and they work great at room temperatures. Us, as the captains of these ships, use radios to send precise navigation information. In response, thousands to millions of NV centers act like tiny compasses. They emit red fluorescence that encodes information of subtle waves of magnetic fields, much like flares with varying brightness levels, guiding us through the sea of invisible waves. My research revolves around one simple question. What if the radio waves becomes inaccessible, and now these NV center compasses stop responding? For example, the radio waves could have been disrupted by a fierce storm, knocking the antenna off the ship, rendering us lost at sea. My project looked at these special magnetic field alignments that unify the energies of different NV centers, even when the radio waves are inaccessible. Now, with the energy of these compasses aligned, they start to interact with each other much more strongly. The result? These NV centers reach a relaxed state faster than normal, and the flares they send become dimmer. So, I apply varying magnetic fields to the diamond and observe the brightness of these red fluorescence flares blinking in star-like patterns, serving as beacons for navigation. This allows us, the captains, to still precisely navigate the waves of magnetic fields, even when the radio waves are inaccessible. Quantum sensing are promising to measure biological signals to help us to perform better disease diagnosis or provide precise navigation even without GPS. However, as the technology is not ready yet to be presented beyond the lab space. So the implications of my research are to make these quantum diamond ships more compact and energy efficient, enabling them to navigate extreme environments where disruptive storms like high radiation or high pressure are present. But we're not just the captains of these diamond ships, we're the captains of our own lives, navigating a world of technology with potential to be improved through quantum sensing. So I strive to bring these sensors to the forefront, ultimately making them ubiquitous in our lives by integrating them into everyday technologies, untethered from radio. Thank you.
Thank you, Chen. I especially like to be the captains of your own life. Good advice for anyone. We are grateful to have representing kinesiology, cognitive motor neuroscience, a field of study, Kirsten Zhang. And what I love is that you're going to talk about brainwaves in basketball. Seems season specific too, which is great.
Yeah.
The neuroscience behind mental skills for peak performance. And there were people a few days ago in the draft who would have been like, I'm going to need this.
We'll talk about that. All right. What if I told you that an athlete sinking a game-winning shot, a surgeon performing a delicate operation, or you giving an important presentation, much like this one, all have something in common? In each case, the human is a performer executing a skilled task under pressure. Many of us want to know the secret to peak performance. How do we perform at our absolute best when it matters most and do so consistently? Cognitive motor neuroscience explores this at the level of the brain. Performance isn't just about physical ability, it's about mental preparation. High performers use mental skills, one of which is called mental imagery, the act of vividly picturing success, to sharpen skills, boost confidence, and control nerves. Prior research shows that physical practice refines the brain for efficiency, making movement smooth and automatic and reducing overthinking. But can mental practice do the same? Can mentally rehearsing a challenging task ahead or a skilled movement also lead to neural efficiency in the brain and resulting improved performance? That's what my research explored, how mental imagery changes the brain and whether training the mind improves real-world performance. I conducted a study using basketball players, and I separated them into 2 groups. One group engaged in mental imagery training, visualizing successful free throws, while the other group did nothing extra. I measured their brain activity using electroencephalography, or EEG, tracking efficiency, and assessed confidence and anxiety, 2 key psychological factors. So what did I find? A single session mental imagery intervention did not change brain activity or improve performance compared to control. However, imagery was associated with better performance, particularly when confidence was high. This suggests that mental imagery enhances confidence, or while it enhances confidence, its effectiveness in improving performance may be highly dependent on individual psychological states. What does this actually mean? Mental imagery primes the brain for success, but we may need longer training periods or a mix of mental and physical practice to see true changes. Future research will give us more insight. Now, this research matters far beyond basketball. Mental imagery is not just for athletes. We all perform every day, whether it is Stephen Curry breaking an NBA nightly point record or you preparing for an important meeting. Performance research applies to all of us. So next time you're preparing for a critical moment, a free throw, a job interview, or a speech, ask yourself, are you just practicing physically, or are you also practicing mentally to leverage the power of your brain? Thank you.
You did see Maryland today. We had a player get drafted, right? And she's a great player. Did you help on that?
We were in Maryland today as well, actually. So, yeah, there's a story there.
Look at that. We got all the winners. Our final presenter, and the font of knowledge that will help us take it home tonight when we talk to friends and family. Let me do this again. Shantun D Vachani is in mechanical engineering. We've been well represented by the engineering school, bravo. Improving electronic cooling efforts through computer simulations is the topic of conversation. Thank you so much.
Fever, we've all experienced its effects. Usually we take a medicine and apply cold compress on the head. So what exactly is the cold compress doing? It's taking heat from the body and reducing the temperature. We can think along the same lines for an electronic device. The rapid movement of electrons produces heat and increases the temperature. Much like us, these devices work well only under certain temperature limits, and therefore there is a need to cool these devices. In high energy data centers, this is done by subjecting the server racks to cold refrigerant-like fluids. So you pass these refrigerant-like fluids over the server racks, and they extract heat and keep the server racks at a prescribed temperature. Because the energy density of these server racks is quite high, the refrigerant starts to boil. So with this as a starting point, I wanted to study the process of boiling and how we can increase the efficiency of this process. First, to save cost, which I know we all like. Secondly, we can drastically reduce the energy consumption, which is a key to sustainable future. So the first parameter that I looked at is, what if I change the nature of the surface where boiling happens? So if you look at the top, this is sort of the boiling that you're familiar with. So I changed the surface and I created a composite surface made of 2 different materials and attached it in a pattern. And so you see that on the right. And you can see that the boiling behavior has changed significantly. But more importantly, the second surface is 2 times as more effective at extracting heat. And that is good. The next thing I looked at is what type of fluid we are using, because different vendors will have different fluids available to them, and how these different fluids engage in the boiling process. Finally, what about space electronics? Experiments in low gravity are very hard to come by. So what I did was I conducted simulations in low gravity and see how the boiling plays out. So finally, what I plan to do is extract all the data from the computer simulations and develop a physical model so that future researchers don't have to do all of this research again. They can just input their parameters, and the model will spit out what is the appropriate surface design for their application. And in this way, I'm using computer simulations and leveraging them for developing transformative cooling technologies. Thank you.
We will think of you in August in this area. We'll be like, oh, we need that cooling technology. One, gratitude to all of our judges who are now doing the hard work of trying to figure out, of all these amazing presentations, how do you think of what is the winners of that? Secondly, thank you to the grad school for hosting this, because we are better for having heard all of the scholarship, but also I think the opportunity to be able to figure out how to do these presentations is worthy. Because someday, whether you're influencing people as a physician with Taylor Swift or figuring out how to make kindergartners want to be teachers, you're going to be talking about something that means a lot to you. And how do you think about that in a very short time to impress people with your findings or your research? The other thing I want to thank all of you for coming. There is something about supportive scholars, but their supportive friends make it possible for them to do their scholarship. So whether you're a faculty member or a friend or someone who's taking pictures when you get up in front of your screen, I think having people here who care about you matters a lot. And finally, you know that you worked hard to get to this place. You were a finalist already, and you'd already done this more than once. And so thank you so much for putting yourself out there and for being in a competitive spirit, because the University of Maryland will rely on you, and you can visualize it in advance, and you did it today. Friday, big day. We're going to find out everything. And it'll be on that 3MT site, right? And so you'll know all about it. Somebody will be $1,000 richer, right? And they can take us for ice cream. But for now, is there some leftover ice cream still? Oh, yeah. No one's having to buy the ice cream today if you get there first. Thank you so much for all of you. And may you continue to have great luck in the world of learning, and go forth and teach us what you know. Thanks a bundle.